Hello, welcome to Progressive Education Society's Modern College of Pharmacy, Nigadi, Pune. In the e-lecture series of in the subject Natural Drug Technology.
Hello, myself Dr. Bhushan Pimple. In this video, we will have a look at cultivation, harvesting, and storage of various crude drugs used for extraction or isolation of phytoconstituents. Specifically, we will be focusing upon the problems associated with cultivation, harvesting, and storage, as well as the various methods or types of cultivation, harvesting, and storage. There are currently various different problems those are associated during cultivation of various crude drugs, and these are the why reasons why a farmer gets low produce as well as low income out of the cultivation. Usually, as far as industrial production of phytoconstituent or extraction is concerned, what industry does is it allots a supplier or it gives the requirement to a supplier. Now, a supplier is not necessarily a farmer, but he is a middleman or an agent. Now, this supplier procures the raw material or crude drugs from either a middleman or a farmer. And if it is a middleman, he in turn gets the produce or collects the raw material from the farmer. Now, in this entire supply chain, there are problems like misidentification or suppose adulteration, unintentional or intentional adulteration. So, due to this, the finished product or the final product of the company is of substandard quality. Now that can be intentional or unintentional. Now we will just focus upon various aspects related to the problems dealt during procurement of material through the supply chain. Medicinal plants, generally, since this is a biological entity, it is susceptible for uh, deterioration or degradation, as it is not a synthetically isolated powder. But since this medicinal plants, they do have certain starch content and all, so there are chances that they may absorb moisture in humid condition and can favor microbial um, growth. Basically, herbal drugs or during extraction, the industry faces a major problem or issue, and that is of quality and quantity of the phytoconstituent. This is not consistent with the batch because the crude drugs, uh, the season in which they are isolated, the pesticides what are sprayed or fertilizers or the geographical condition, everything matters a lot during the quality and quantity of phytoconstituent. As far as quality and quantity of phytoconstituents are concerned, this varies throughout the year. Adulteration. As we have seen that there are chances of adulteration, or sometimes you can call it as misidentification of the plant, or a look-alike plant can be procured uh, by the farmer or a middleman, and this can cause a serious threat to the industry as well as its products. Also, a collection of wrong part of the plant. Usually, what happens is the industry gives a contract to a person, and the person gives in turn the contract to somebody else. So now he just lists the name of the plant. Many a times it happens that the name is correct, but the part is sometimes missing in the list. Part of the plant is missing, and so for that reason, the entire plant is collected. We will take a very simple example of Trigonella phenum, that is methi. In that case, the seeds are usually required for an industry to separate or isolate trigonelline. And sometimes there are chances that the middleman can procure or get the leaves of trigonella, and the leaves have very less amount of trigonella. Very similarly, if seeds are concerned, let us take an example of Nux vomica. Nux vomica seeds, if you read the entire biological source, it says it consists of dried and ripe seeds. Now, many a times the ripe seeds and unripe seeds both are mixed together by the collectors, because Nux vomica is not cultivated by the farmer, instead it is collected from wild sources, from the tribal people. So there is a method to distinguish between ripe and unripe seeds. The seeds are soaked in water tank, and the seeds those are floating on the surface are discarded because they have more amount of oil content and they are immature. So such immature seeds do not have more amount of strychnine or brucine. Likewise, the part that is to be collected should be uh, perfectly chosen and then supplied. Then about microbial load. Microbial load means here during collection, there are chances that the part or the plant itself has attracted fungus, or can be loaded with fungus or other microbial spores. So these fungal spores during transportation can spread the disease throughout the produce, or it during the storage also it can spread the disease or spread the fungal infection. Now, the presence of microbes can cause isolation of their toxins along with the phytoconstituents. So, sometimes the separation of phytoconstituent from these toxins becomes difficult, or sometimes the toxin remains unnoticed and is carried up to the finished product. Adhered impurities also are very important as far as the weight of the product is concerned. Suppose generally this is the case with roots. Roots are having, or they are adhered with soil, and due to this their acid insoluble ash increases. So now this is a very critical step to determine the acid insoluble ash before allowing the crude drug to for processing. So this lets us identify whether the crude drug has been containing huge amount of inorganic material, or it is starch. Generally, the crude drugs they do have starch in them, like especially tubers or roots. They have starch, and this starch, uh, in presence of humid or moist environment, can further proliferate the fungal growth. The way how the crude drugs are stored or transported is very important, because during transportation, if it is rainy season, there are chances that the moisture may get lobbed or trapped into the crude drug, and this can increase the rate of degradation of the crude drug. Harvesting should be done especially by trained workers. Unskilled workers can cause damage or cuts or crevices into the crude drug, wherein soil or soil-borne bacteria can house. Storage of the crude drug, generally all the crude drugs are stored 1 or 2 feet above the surface of the ground, because there are chances that rodents or insects, crawling insects, they may attack the crude drug, or sometimes even the water, if it is lodged into the storehouse, there in that case it may cause problem to the crude drug, even during handling or during handling or the uh, of the crude drug or the powder may get spill onto the floor, so that has to be cleaned properly. So storage as far as crude drugs are concerned should be done in plastic containers only if they are completely dried. Now we will have a look at the factors that directly affect the quality as well as quantity of phytoconstituents in the crude drug. Geographical location. Now we will take a very simple example of strawberries. Strawberries require cold climate, but not a frosty climate. That is the reason why strawberries cannot be grown in Kashmir, but they can be very well grown at Mahabaleshwar region because of the exact temperature required. Even it cannot be grown in humid or hot climate, arid climate. So, depending upon the crude drug or depending upon the growth or of the phytoconstituent, the ideal place for cultivation needs to be chosen first. Second, climatic condition. Climatic condition has a direct effect. Like say suppose in harsh sunlight, there are chances that certain phytoconstituents they get photo-oxidized. So most of the plants cannot survive or tolerate summer or dry areas, whereas others may not be able to tolerate humid environment or rainy season to a large extent. So that is the reason why collection or cultivation should be done at appropriate season. Sometimes you will notice that cloudy atmosphere can reduce the flowers or flowering of the plant. Now, since flowerings are reduced, that means the fruits will be reduced. The number of fruits will be reduced. So, depending upon the crude drug, the climate that is suitable for the crude drug should be chosen for cultivation as well as harvesting. Generally, during harvesting, rainy season is generally avoided because we will take an example of barks. Are generally not separated during rainy season, or even fruits for that matter, like mango, cannot be harvested during rainy season because that is not the appropriate season for. Next, quality of the crop. Quality of the crop means the breed, or you can say the genetic authenticity or genetic makeup of the crop. Many a times we read in the newspapers that the farmers they are unable to produce because of lack of low or because of the low quality of seed that is provided by the government, or sometimes the seeds are unable to germinate. So quality of the crop, that means whether it is of a proper genetic makeup, that directly influences the phytoconstituent yield and quality. Then type of soil. Soil varies with the geographical location as well as during cultivation. Soil is added externally sometimes in a small piece of land. Now the quality of soil you are using can be of large granular size called as loamy soil, or very fine soil called as sandy, or sometimes clay soil. Now, the particle size of the soil, it affects the irrigation. That means water, if the particle size of the soil is too less, water remains lodged near the surface. So if you are interested in roots, the roots are going beneath the soil up to certain meters, and if you want the water to go up to the roots, the soil chosen should be of larger particle size. Whereas for advantageous root containing crops, the clay soil is ideal because it allows the soil to remain in the allows the water to remain in the superficial layer of the soil. Next, irrigation. There are various modes of irrigation. Generally, most of the farmers they depend upon nature for irrigation, and accordingly they choose the cultivation type and crop for cultivation. But if if there is considerable amount of water, in that case the farmer can opt for various types of irrigation. Now, it is a very wrong concept that more water will produce more good quality of crop. No, it is not so. The type of irrigation varies. Sometimes the adjacent irrigation, that means drains, or sometimes overhead irrigation, also called as surface irrigation, or sometimes drip irrigation, is facilitated. Now, overhead irrigation has several advantages as well as disadvantages. There are chances that the moisture may remain on top of the leaves and can cause degradation to the leaves in case of surface irrigation. And over irrigation can also leach out the fertilizer or nutrients from the soil, and it can take the nutrients to the water table, or to away from the plant and below in the soil. Then fertilizers and pesticides used should be of optimum quality. It is not that more fertilizer will produce more good quality of crop. No, because sometimes the micronutrients are to be in sufficient quantity. Micronutrients in case for example, like magnesium, manganese, cobalt, iron. These are micronutrients, or sulfur, phosphorus. These are to be supplied in appropriate quantity. If they are more, there are chances that other plants may survive or grow by consuming these fertilizers. Now, that can increase the yield of unnecessary or unwanted plants. Age of the plant. While harvesting the crop, the age of the plant is very, very important. We will take an example of ephedra. Ephedra is a stem, and the biological source for ephedrine or pseudoephedrine, the quantity of these alkaloids is more in young stems. As long as the stems that are collected are young, that means greenish in color, they have more amount of ephedrine, pseudoephedrine, and norephedrine, other like alkaloids. Whereas if it is collected, if the ephedra stems are matured, they have some bark over it or are brownish in color, such ephedra have very less amount of ephedrine and ephedra alkaloids. Similarly, the fruits those are collected should should have a specific analysis of the phytoconstituent. Before collection of the fruits, the optimum quantity of phytoconstituent should be analyzed first, and then those should be harvested. um, there are certain plants like vinca also, wherein vinca entire plant can be used, but if the leaves are too small, the quantity of vincristine is too less, so the leaf size has to be the plant has to grow, allowed to be grown up to a certain height so as to develop proper size or size of the leaves, and then only appropriate amount of vincristine and vinblastine can be isolated from it. Time of harvesting, as we have seen that rainy season is generally avoided for harvesting of most of the crude drugs. Further, the Analysis of the crude drug has to be performed to determine the appropriate time of harvesting. We will take an example of opium alkaloids. Opium is a latex that is obtained from Papaver somniferum. Now, this latex is mostly secreted in the morning. So, incisions on the fruit, vertical incisions on the fruit are given in the morning so that the latex oozes out and it keeps on oozing throughout the day, and in the cold weather of the night it freezes. So the frozen latex is then scraped off in the next morning. Similarly, tea leaves have more amount of flubafine and tannin components if they are collected in the morning. Furthermore, the size of the leaf, tea leaf, is very, very important. The top three leaves, the topmost three young leaves, they have more amount of green tea oil compared to the matured leaves, so that that is the reason why the tea leaves are hand picked by the workers and those are collected. Such kind of tea leaves will produce a good quality of tea, whereas nowadays hand picking is abolished in certain area and instead those are directly cut by long scissors, and this gives rise to fine dust tea or the tea that is stronger but does not but lacks the aroma of green tea oil. Part of the plant harvested should be very the harvester should be careful during collecting the part of the plant, because if the harvester is unaware as to which part has more amount of phytoconstituent, in that case a wrong part will be supplied to the industry for isolation of phytoconstituent. Storage condition, as we have discussed in the earlier slide, has a direct impact on the phytoconstituent, because there are most of the phytoconstituents which increase or change to their metabolite form during storage. Those are called as post-harvest physiology. We will take a very simple example of grains. Generally, the rice or wheat grain, those are chosen that is based upon the aging of the grains. More the rice or wheat are aged, their quality increases, their aroma increases. So this is the reason why post-harvest physiology plays a very direct impact on the quality of the plant metabolite. Then storage period. Sometimes on storage, the degradation of the phytoconstituent occurs, whereas sometimes for other crops, storage can facilitate the formation of a particular desired phytoconstituent. Uh, like, so suppose red chilies, if you store them for a longer duration, the redness increases. If you dry them properly, the redness increases. Whereas cloves or black pepper, if you store them for a longer duration, since they have volatile oil in them, slowly the volatile oil diffuses out and the crude drug will be over the period of time exhausted of the volatile oil. It will have very less amount or negligible amount of volatile oil in them. So storage period is also has to be determined first. Microbial contamination sometimes causes change in the phytoconstituent. It can add its metabolite and thereby inhibit certain biosynthetic pathways, or sometimes can facilitate biosynthetic pathways. Now about cultivation. Now exactly what is cultivation? It is the act of caring or raising the plants. It is very similar to raising pet animals. Cultivation is raising the plants. Harvesting, it is the process of collection or gathering of ripe crops from the field. It is very necessary if you are collecting the crop only after sowing. That means first you are sowing the seeds or the seedlings and then you are collecting them. Such kind of collection is called as harvesting. That means harvesting is the term used for collecting the crop which is artificially sown or artificially cultivated or grown. And if you are collecting it from wild sources, say for example timber or naxwamika seed, in that case harvesting is not used. In that case collection is the term. Uh, collection is the term used for collecting the raw material from wild sources which you have not artificially sown. Steps involved in cultivation are like suppose preparation of soil or first site selection. Ideal site should be selected for cultivation of the desired crop material, and this can be done by thorough literature survey or from gathering the experiences from various farmers who are engaged in cultivation of the same plant or crop. Second, selection of the season. The right season has to be sorted and selected for sowing the plant or collection of the plant. Soil selection. Soil, depending upon the plant part that is to be collected, should be chosen like sandy soil, sandy loam soil, or loamy soil, or sometimes the mixture of these. Then preparation of the land. This is called as proper addition of soil, the layer of soils, or addition of certain fertilizers, or addition of sometimes ash into the soil. Now this, there are various techniques called as tillage technique. In tillage, we uh, change the soil. That means the lower soil is taken on the upper side and the upper soil is pushed at the lower side. This can be done by ploughing, or it can be done by with the help of tractors or mechanical agitators. Here the soil is agitated and it is made upside down so that the lower nutrients they come at the surface. Then mulching. Mulching is the process wherein the evaporation of water from the soil is avoided. Nowadays, you might have seen many a times in the fields that plastic sheets are placed over the surface of the soil. Now, this plastic sheet is one kind of mulching. Initially, or in a traditional ways, certain sand or um, pebbles or stones were placed. Sometimes even wood chips used to be placed to avoid the soil from getting completely dried or prevent the water evaporation from the soil. Water evaporation not only makes the soil dry, but also the fertilizers are locked in the state. That means they cannot be transferred towards the soil unless the soil has water into it. So, mulching is necessary. Sowing. Sowing can be done by various means like broadcasting the seed or placing the seeds into the dug small pits and other ways. Watering, that is irrigation. Irrigation, the water quantity and the quality of water changes with the age of the plant. Fertilization is the next step. Once you have sown the seed, watered them, then once the seedlings come out, fertilization is generally initiated. Weeding. Unwanted plants are called as weeds. Now these weeds, they take up the nourishment or nutrients of the desired crop. So weeding is to be done. There are various weedicides, or generally those are picked up manually or by mechanical weed harvesters. Then pesticides are sprayed. Pesticides prevent the growth of certain insects or even certain weeds, weedicides. Even rodents do not attack the crop if pesticides are sprayed in a proper manner. Harvesting. Once the crop reaches a desired state, harvesting, that is collection, is initiated. After harvesting, the crop has to be sorted. Sorted means there are various ways of sorting. Sorting means separation of the desired plant part. You might have seen beating or threshing. Threshing means here the entire dry plant is uprooted and then it is beaten against wood or a stone so as to separate the seed. That is the one method of sorting. Sometimes manually or by a simple hand picking, sorting of the desired plant is done. Lastly, storage. Storage is to be done according to the requirement of the plant. That means sometimes it is directly stored. If these are fruits, those are cold stored in the cold storage, or sometimes if they require certain drying, then processing is to be done and then they are stored and delivered to the desired location. There are various methods of cultivation. You might have seen in your earlier school days or college days the various methods of cultivation, like sexual mode of reproduction or sexual method of cultivation, wherein seeds are propagated or the plant is cultivated by from the seeds. The plants are raised from the seeds. A seedling is the crop or the plant that has developed directly from the seed. Seeds are sown either by using broadcasting method or by dribbling method, or placing them in drills or holes which are manually made. The seeds should be of good quality so as to get a considerable amount of germination rate, and should also be free from certain diseases or uh, disadvantages. There are certain disadvantages associated with this method, like seedlings obtained, they require some more time to bear fruits. The reason for this is more amount of time is engaged to develop or for germination. It takes sometimes 5 days or 15 days for a seed to germinate. Later, after 15 days, it will develop into a seedling which is up to a height of 1 meter, or sorry, 1 feet. Now that will take some extra time of 1 month or so. So the entire 1 month or 2 months are wasted, and thereafter the plant bears fruit. Other disadvantages are like non-uniformity in their growth. Not all the seeds will grow at the same rate. Some seeds, they even though they are placed or they are sown on the same day, the growth rate varies, and the crop during the harvesting time, you will notice that all the crops are not showing the expected growth rate. Even the yield, it varies from plant to plant. In a same field or in a same piece of land, the yield varies from crop to crop. That this is because of the genetic variation. Since it is a sexual mode of reproduction, The genotype varies, and therefore the phenotype also varies. You might have studied about Mendel's experiment of pea plant. In the same, very similarly, here also yield varies according to the genotype. Then the cost involved in harvesting and protection of the plant right from its seed. Generally, when you sow the seed, you will notice that there are many birds in your field that generally they feed upon the sown seeds. So it requires manpower to protect the sown seeds from the migrating birds and also the seedling from various pests. Another mode of cultivation is asexual mode of reproduction, which is also known as vegetative propagation. Here, vegetative part means non-reproductive part. The reproductive part in the plant is only flower. Now, suppose you are developing a new plant from a non-reproductive part, like suppose leaf or stem or root, then in that case it is called as vegetative propagation. Examples for this is bulbs like squill, garlic, or sometimes onion, but onion is very rarely cultivated by using bulbs, but garlic and squill are cultivated with this method. Corms are underground stems of the plant, and these for in this way, colchicum and saffron are cultivated. Tubers like jalap, aconite, or potato, those are sown directly, and from them the plant can be generated. Rhizomes of ginger and turmeric are utilized or conserved and then utilized for plant propagation. Runners are the plants that spread above the surface of the soil and they travel a certain distance, like, for example, peppermint or um, mint or even strawberry. They are cultivated with the help of runners. Then suckers like mint, pineapple, or banana, these are the help of suckers. So these were the methods of cultivation. Now next are method of harvesting. Once the crop is ready or the plant of the part is ready for harvesting, various modes of harvesting can be followed. Now harvesting depends upon the part of the plant. If the part of the plant is a fruit, then sometimes those can be hand picked with the help of bamboos or certain mechanical sticks which have the two-way finger like clamp, and through this the unripe fruits are taken out, picked from the plant. Then in case of olives, thrashers or shakers are used, wherein the plants are shaken with mechanical shakers, and the loosely held fruits they fall off. And to prevent the fallen fruits from damage, those are collected in large sheets, cloth sheets. Then the certain mechanical harvesters are there, generally for harvesting the sugarcane or the potatoes. In most of the European and US countries, mechanical harvesters are utilized. As far as barks are concerned, the barks can also be harvested by various methods, like giving a simple incision, vertical incisions, two vertical incision and a horizontal incision. The bark can be stripped off. The bark can be separated into small flakes or sometimes large strips. So this method of harvesting is called as stripping method. Second method is felling and girdling. The entire plant is cut. The stem, large stems are cut at the base, and the stem or the wood is separated by giving a circular incision across the stem on both the surfaces, on both the sides, and a central vertical incision is given so as to separate the entire bark. One more method is girdling method, wherein the bark is collected in a horizontal manner in this case. So these are the various modes of harvesting. Then methods and steps involved in storage. Drying is very critical step before storage because the drying increases shelf life as well as it decreases the microbial contamination, and it also aids in grinding. A dried crude drug can be ground much more efficiently, and the particle size can also be controlled in that case. Drying also inhibits enzymatic processes. Enzyme activity is also inhibited by drying. There are various modes of drying, like slow drying is preferred in case of vanilla pods. Vanilla pods are containing sticky material which has the vanillin into it, but it requires enzymatic activity. So to continue the process of enzymatic activity, the drying rate should be at a slower rate. Drying should be done at a slower rate. Whereas in case of chili and carrot, the drying is to be done at a very fast rate so as to inhibit the enzymatic activity, or else the chili and carrot gets degraded. Various types of drying are like natural drying, which makes use of sun drying. Directly, sun drying is preferred in case of chili, and shade drying or air drying is preferred in case of um, volatile oil containing drugs like pepper or cloves. Artificial drying nowadays is carried out in order to increase the rate of or the speed of drying. Herein, tray dryers are used or vacuum dryers are used. Spray dryers are used only in case of extracts and not crude drugs. Methods of drying. Directed drying, the entire crude drug is without cutting, the entire crude drug is directly subjected for the process of drying. Like, for example, berries, as we can see in case of resin, the development of resin, that is kishmish, and chilies and leaves, those are directly dried. Slicing and drying. Slicing or cutting, it increases the surface area and then it facilitates the drying process. Further, certain cracks and crevices which holds the fungus or certain microbial contamination can be exposed directly to the sunlight, and their contamination can be prevented or can be minimized. Then fermentation and drying. Fermentation and drying process is carried out in certain crude drugs, like for example tea. The tea leaves are taken; those are then soaked in water for certain time, and those are fermented. And the fermented part is then dried, and then it is powdered. Fermentation converts the flavonoids into tannins as far as tea is concerned. Other steps in before the drying are garbling. Garbling is removing of the sand or adhered soil or dust, stones or foreign organic matter like animal excreta from the uh, crude drugs so as to minimize the microbial contamination or minimize the adulteration, unintentional adulteration. Packaging. This is the third stage, that is during before drying. Sometimes the moisture sensitive material like powder of the barks, these are to be stored in airtight container and away from humid atmosphere. Generally plastic containers are used. Light sensitive material needs to be stored in opaque container like plastic containers of or plastic drums so as to avoid photo oxidation. Then there are certain thermosensitive materials which are to be like suppose fruits. So these are to be stored in cold storage condition and thermosealed so as to maintain the cold atmosphere even after even if the material is taken out of the cold storage. Then pressure sensitive material which can uh, cause which are very brittle and can be subjected to formation of powder during storage or pressure are to be stored again in hard plastic containers or wooden containers. Certain adsorbents are to be added to the airtight container in order to absorb the moisture from the crude drug and prevent the or avoid microbial contamination. Moist materials like say suppose onions or garlics are to be are can they cannot be dried directly in sun, so that they are stored in gunny bags and ventilated containers so that slow drying is facilitated. There are various types of storage, like cold storage. This is usually required for fruits or fruit juices. Then a CO2 rich environment is given to the fruits or certain materials that immediately ripens. The CO2 it acts as an inhibitory enzyme or inhibitory material. In high CO2 content, the enzymatic activity stops, so the fruits remain in unripe condition. So during this is usually done, so suppose if you want to export the crude drug and which to countries which are very farther and through the sea route, so it takes sometimes 15 days or 1 month. The fruit when it reaches the ports, it is completely degraded. So as to preserve its nutrients or to preserve its unripe condition, the fruits are stored in CO2 rich environment. Ethylene oxide, it facilitates the ripening process. So this is usually done in case of banana and mango, wherein ethylene oxide is sprayed and or ethylene or it is stored in ethylene oxide gas, and this is aging or growth hormone, so it facilitates the ripening process. Even certain crude drugs they require warm and dry storage, as is the case in mango, wherein if you store them unripe mangoes in warm and dry climate or condition, the ripening process is initiated. So depending upon the place where the material is to be transported, the dependent storage is chosen accordingly. So this is all about the methods and steps in the part 1 of this cultivation, harvesting and storage.